Kamal sir, one of India's Ahmed Muslim University. After completing his doctoral studies, he joined CSIR IISc Hyderabad as a scientist, where he made significant contributions to bioorganic and medicinal chemistry research. He further enriched his research experience through postdoctoral work at the University of Portsmouth, England, and the University of Alberta, Canada. His significant contributions provided him the opportunity to lead the establishment of ANRF-PAIR Hyderabad, which all of us are familiar, and later served as director and outstanding scientist of CSIR IISc Hyderabad. Professor Kamal played an instrumental role in establishing the Biotechnology Incubation Center at the Biotech Park in Hyderabad, and also contributed to the conceptualization and establishment of several important initiatives, including the National Center for Research and Development in Bulk Drugs. So 115 PhD scholars, reflecting his profound impact on medicinal chemistry research and scientific leadership in India. He is the recipient of several prestigious recognitions, including the CSIR Young Scientist Award, Grand Bax Research Award, OPPI Scientist Award, and fellowships of the National Academy of Sciences, India, and the Royal Society of Chemistry. Today we are fortunate to have you, sir, here to deliver a lecture on design and development of potential anti-cancer agents. With great respect and pleasure. Thank you, sir.
So once again, welcome back on one specific topic. And these are again scaffolds from nature, but the total synthesis have been carried out so that you can carry out the structural modifications and so on and so forth. Can you see the slides, or the lights are too much? This is just a brief outline of how the medicinal chemistry got evolved. So people, when there were problems, there were diseases centuries back, and people had to find out how they can treat them. So in those days, many things happened, and practically the Ayurveda developed during that period, and Egyptian medicines, what we call them as Babylonian medicines, Chinese medicines, most of these things have come under the ancient period. And again, something came back in the medieval period, what we call them as Unani medicines, medieval medicines. These are mostly, they traverse through Greece. That's how the name Unani came, through the Middle Eastern countries, even up to India. So that's the type of medicines. Then in the modern one, mostly the medicines were developed by purifying, getting single molecules, particularly from natural products. See, as in case of Ayurveda, Unani and all that, we know very well that they are practically the actual plant product comprising of several ingredients, compounds, molecules, and that. So they call that as a natural formulation. But in the modern medicine, one needs to look into a specific molecule which has activity. So that's how these things got. And what we see now is the current scenario. The most important thing is what we have been talking also in the projects and programs now, the rational drug design. This is where is the totally different aspect. And this rational drug design could be through various ways. And again, the spontaneity of processes with high throughput screening, nanomedicines, particularly in the drug delivery aspects, targeted therapies, and all that has come into force. So going through all this, we see that the programs, what we are trying to do now in the ANRF-PAIR, as well as the project, what we will be doing in my, the ANRF Prime Minister professorship is also something on those lines with specific targets and so on and so forth. So what you see here, this is where we are, and we are looking for speed again. How do we do the whole process? As mentioned, the whole process of today drug delivery, drug design discovery happens up to something like minimum of 12 to 15 years. And there are some shortcuts for some emergency ones, particularly in cancer and AIDS and all that, where you can probably complete within 9 to 10 years, the whole program, once you have the actual new chemical entity, the NCC. So let me now go through a specific example on which we worked, I would say, extensively. This is basically a scaffold, what we call it as pyrrolobenzodiazepine. So it's a tricyclic ring system. And practically, this is, this class of compounds, they were all isolated from Streptomyces species. So practically, they are natural. And all these new molecules, I could say from tobramycin to chicamycin to anthromycin, most of them have all gone into clinic also at different stages. In fact, tobramycin has gone up to phase 2 clinical trials, and even anthromycin has as anti-cancer drugs. So this is where a problem happened, that these molecules were very good with respect to their biological effects, but they had severe cardiotoxic issues. So then a couple of groups started working on these natural products, utilizing them as a scaffold, and then designing new ones. How can we reduce the toxicity and retaining or improving the actual effects of the molecule? So this is where some programs started, and there were hardly 2 or 3 groups working in the world. And then we also became one of them. Here, when we talk of a molecule, when we looking for developing or discovering something, you need to understand how the actual compound works in the biological system, what we call it as something like a mechanism aspect. Here, for example, most of you, just for the students, you know that these molecules are supposed to be DNA interactive, and particularly they basically get inside the minor groove of the DNA. And after getting inside this, what happens is they form a covalent linkage with the immunofunctionality. With this immunofunctionality, with the NH2 of guanine. You'll come to know in the forthcoming slides. This is what was considered as the proposed mechanism, but basically it was validated through mostly the DNA duct formation and all that, through a lot of NMR and many other detailed studies. So everything was confirmed on this. The only thing I would like to mention is this duct formation makes it to, it's a cell kill aspect which happens once it enters the minor groove of DNA. Now, when you're looking for such compounds, the other important thing one has to see is there is a molecular recognition of DNA aspect involved in this. And that is, this particular molecule has a preference for purine, guanine, purine base pairs. Why this is important is, because many a times, most anti-cancer compounds are highly, not only, cytotoxic to the cancer cells, but they are toxic to the normal cells. So what was believed, that the more you can have sequence selectivity, you can improve upon its toxicity. That is what, so that they can differentiate to an extent between the normal cells and the cancerous cells. So keeping that in mind, one looked into this. This is just to show you how a compound like anthromycin or tobamycin fits so well inside the minor groove of DNA. It's just a representative one. Now, when you are looking for making molecules, you need to have a proper synthetic strategy. It should be a synthetic strategy which is practically viable, so that you can get enough compounds when you want to evaluate them and eventually to become a drug tomorrow. So those synthetic strategies, you need to have, mastering has to be done in a way. And for that, through the retro of that tricyclic ring, you can see that these type of scaffolds can be used as precursors for the synthesis or the total synthesis of such molecules. So these are a couple of reported procedures which were already there in the literature when we were starting on this journey. And this is a Kaneko's approach. You can see very well how this whole process happens to get finally the imine formation of this. Because you will come to know why this imine is really required. As I mentioned to you, it forms with the NH2, it has to form an adduct in the DNA. This is another method which is called as Thurston's approach, which this group has worked on this. Again, there is a little difference in this, where you have again the reductive ones, reductive cyclization and so on and so forth. I'll just run through because I'm told that I have to finish much early. And then you have another Fukuyama's approach, where you get through again protective, deprotective aspects in this cyclization process to finally get this imine. Then we have also developed now a couple of, because what we observed was in all those methods which were reported, there were variations. When you have substitution variations in the scaffolds, all the methods were not working alike. So we wanted to generate more synthetic strategies so that it becomes much more common for various substituents we want to get in. You'll appreciate when I talk about how this aspect happens with respect to, because while you are doing this, the most important thing is the imine functionality is a highly reactive functionality. Even the storage becomes very difficult, because if you don't do it properly, you lose that imine. And once you lose that imine, the activity is gone. So in the process, we also developed a large number of, I would say, greener, different type of methodologies, so that we have a large number of options. These are a large number of, again, reductive cyclization approaches we have studied with various agents, how they function. I can run through because I think this button is giving me problem, Raju. I don't know why. Okay, maybe it's okay. You don't have another option, or? Let me try otherwise, but let me see. Now, see, when we were doing, in those days, most of medicinal chemistry, solid phase synthesis was becoming very popular, because we had mostly many laboratories, industry, they had acquired autosynthesizers, where you can have at a time large number of molecules synthesized, so that you don't have to, again, that just do purifications to an extent and then go for the biological evaluation. So that way solid synthesis thing became quite popular, and that's how we developed some strategies for these molecules, and a large number of libraries were also developed in that. Then this was again to keep something with respect to greener, with respect to the reagents. So that's how we have polymer supported reagents to come to these molecules. Then these are a couple of examples where click chemistry was also implied while we make dimers. I will talk to you about the dimers shortly, how they are very important with what we call them as when you have this benzene to benzene, they are called head-to-head dimers. And when you have one benzene and the other, the pyrrolidine molecule, subunit, they are called as head-to-tail dimers. So such things were developed when we want to make libraries of such compounds. Now coming to this, which is very important for a medicinal chemist, while you're proceeding on developing something, molecules, you have to have a clear understanding of a structure-activity relationship. This is because you know that which functionalities are very important, which groups are very important for the activity. So you can't anything minus that, you may lose the activity. So keeping that in very briefly, I can tell you, as I mentioned, this imine functionality is most important. And this proton, that is the stereochemistry which sits on the top, is also very important. Because since this molecule has to enter the minor groove of DNA, it has to have a particular shape. And that will happen only when this is on the top. This can happen, I generally give an example of an half-opened envelope lid. So that is how this will look like a lid of an envelope. It's half-opened. So that shape is really important to get inside the minor groove of DNA. So these things have to keep in mind. Because we tried having a racemic compound, we tried even the other isomer, the R form. In racemic, there was a very minimal activity, and R form, there was totally, activity was absent. This is better? Okay, thank you. So now coming to limitations, there are mostly naturally occurring normal cells also, as I mentioned to you. That's how I already mentioned to you that these base pair aspects are very important. So what happens is, in case of monomers, they are the 3 base pairs, which they are having that affinity. But in case of dimers, you're enhancing that 3 to almost 6 base pairs. So that is how the selectivity further improves in case of dimers. So this was basically initiated by one of the group in UK, although again we did a lot of work in such molecules. So these, this particular compound, basically this also went for phase 2 clinical trials, but they had issues of, again, bioavailability. So keeping that in mind, we thought of designing something little different, and that's how we thought of such a molecule. We looked into all aspects involving modeling, so many things. And what we can see the difference from the previous one to this is, see here it's only an alkane chain. It could be 3, 4, 5, 6, depending on various sizes they have been doing. And one specific size really is good for a proper fit in the DNA. But in this particular thing, what you see is, we have brought in this piperazine moiety, because piperazine is known for improving the solubility and bioavailability. Once we brought in this, we examined this, and we could see that it not only improved the bioavailability, but this molecule also improved. See, this is the dimer, what you see, and these are the thermal denaturation results. I will just briefly tell you, and you can see there is a big difference in one of the molecules of this compared to the one which you saw in the previous one, which went into the phase 2. So here, the DNA melting is something most of you may be knowing. It's mostly like the opening, the whole DNA, double helix is practically open, just like a zipper when you open a zip, and the whole thing goes up. So this happens at a specific temperature. And when such molecules are mixed with calf thymus DNA, the calf thymus DNA without the compound and with the compound. So that difference is nothing but the delta Tm. Because what happens is once it forms this, binds to the DNA, the thermal denaturation the melting point of that particular aspect enhances. So that difference is called as thermal denaturation. And we can show that how nicely this goes inside the DNA, even this dimer. So a lot of studies have been done in detail. I won't get into all that because this is all brought out in detail. Now I'm coming to something where we were looking for fluorinated groups, how it would, in some cases what we saw is even fluorination at this ends brought some benefits with respect to the DNA melting. The other one, we can see that here we are trying to change amido aspects. And here, what happens is when such functionalities were brought in, they didn't function very well. In fact, we lost most of the DNA binding aspects. See, compared to the DSB 120, it is hardly much less, almost 50%. Then these were again dimers where different type of chains were brought in with again amido. This also did not give us very good results compared to the actual with just the alkane ones. So, but anyhow, we made some attempts to see whether that would do well, because we thought this amido functionality could also bring some more interactions, because there are a lot of interactions that can happen in the DNA. Now, this is again where you brought in I would say triazolo type bridge also was brought in to bring some interactions, and that is where some of the molecules showed very interesting, these were all designed and activity, but not that great, but definitely much better than the ordinary one. So in those cases, but here I would say the activities of this is for a totally different compound. Now here I would show you. See this, in the case of dimer, if you have seen, they were practically two imine functionalities. So what was claimed, which was developed by the UK group, was that it has a cross-linking for two DNA chains. So that's how the cross-linking was happening, and that's how the melting, the thermal denaturation was increasing. But we wanted to understand that if we have a dimer, but only one imine functionality, how does it behave without two imine functionalities? So that was again interesting to see that this particular molecule without two imines had a better DNA melting profile compared to the one with the two imine functionalities. And we have discussed this and tried to explain this with the other interactions it has other than directly the covalent linkage. So this is for again something improvement somebody can do for making such molecules. Again, you can see some of the in the modeling aspects how these things fit. Now the other thing which I would like to mention is this is again for the first time people worked on only monomers and the dimers, but we wanted to do something different. We thought, why can't we bring the other DNA interactive compounds and bring that to this scaffold which has a covalent linkage. That is the pyrrolobenzodiazepine with other heterocyclics that also are known to interact with DNA, but without covalent linkage. So I would say we have done a huge study on this with hundreds of molecules prepared on these lines from various scaffolds. And this is again, I would say this is another part of, a drug in fact, which has a DNA interactive activity, which was brought in with various substitutions, and again the chain lengths different to this particular. So this is more like the conjugates and the hybrid aspect we initiated in those days. And it's become very popular today in many class of compounds, particularly for the cancer because many of these are now being done for the sake of bringing two types of targets can be involved in a single molecule rather than giving a combination. So such things have become very common today and giving excellent results. So we can see that how nice the things were, much better than a PBD dimer what we had before, and with this type of conjugates the things were far better off. I'm not getting too much into details since I think maybe in another 5 minutes.
10 minutes.
Okay. So these are some of the studies that were done in collaboration with some of our other colleagues, where to see that how such things, where they have this type of complex formations, which type of DNA aspects are involved. So many things were studied in this. So that this can bring more refinement while you're designing the next molecule. That was our main interest. And again, you have a different type of benzothiazole now brought in, benzoxazole we brought in to again link with this pyrrolobenzodiazepine. These also behave extremely well, and a lot of, I think through a lot of modeling aspects, we could refine them to what we really wanted. So they gave. Now this is another thing, naphthalamides, bisnaphthalamides, they are all known previously for as anti-cancer compounds. But the thing is, they're highly toxic. So here we have tried to bring in the combination of these two also. And interestingly, we brought in also, because these are also having issues of bioavailability, most of the naphthalamide derivatives. So we brought in this piperazine also and could get some wonderful compounds. Majority of the compounds had very good DNA binding ability. You can see a very detailed aspect was carried out. How exactly it happens, this was, I think, with one of our German collaborator from University of Greifswald. He was an expert in NMR with DNA, how it behaves. So very detailed, two-dimensional and various detailed NMR studies were carried out to look into all the, what type of base pairs were getting, which type of interacting and where it's getting inserted. All these things were understood. That also helped us in again further designing some molecules. Yeah, that's the whole thing detailed what you have seen for these molecules only. Yeah, so this is again the summary of how all these studies have become useful for us to become, bring new molecules. Now we have again a lot of other combinations. I won't go into very much details, but these are again because it's a very long journey we have traversed on all these molecules, utilizing them for understanding and also to come out with some new molecules. This is again simple modifications with other heterocycles and heterocyclic groups, and what type of other aspects also happen. So we have also studied, apart from the mechanism aspect of DNA, covalent linkage and other interactions, many other enzyme studies we could understand, because these molecules were known for such things. So we had improvements in, again it becomes like dual target type things. So, and also you can see that the activities are very good. They're all less than 0.1 micromolar. And this is again another series where we have done these things. I will rush through something so that again various heterocycles, a lot of detailed studies. I'll just rushing through some of these so that I can complete. Yeah, this is one thing I'll just make a mention. See again, the delivery also is very important when we talk of such cytotoxic drugs which are highly toxic. So we have designed something on the ADAP and the prodrug monotherapy basis. wherein we have tried to link this as a self-immolative spacer, in this case what you see here, and also an enzyme substrate. So this is what we call it as a prodrug. And we want this to be utilized so that in the biological system, it gets cleaved and gives the actual drug. That will be, once the whole thing gets cleaved, you get the immune functionality to function as a drug. So this study was done in detail, and what we could see was a very interesting thing again here, that these particular cleavage can happen where it will get cleaved and and can act selectively at that site. So a couple of animal studies were, I'm not bringing all that in this, was done, and it was extremely interesting. I will just finally talk about some molecules which were licensed to a US company. They are basically, I would say, 5 series of molecules which were selected by them, where all the US, PCT, Indian, most countries' patents were there, and these were all licensed for their preclinical development. These are all the ones. So in summary, this is what it is. I can only say that this is a journey for drug discovery. It may be useful now. How this thing one tries to do by utilizing again the AI/ML. How do you apply that? This is what it need not be just these molecules. It could be any other molecules. So how do you shorten the thing? Although AI/ML will go into the complete development of the drug, the entire process of the drug. It could be from your preclinical to so many things, even understanding the toxicity aspects and so on and so forth. But at least while we are designing, how can we do that? This is what I think the PAIR program will be looking into, and also which we are going to do in our project of ANRF. We will be looking for the dual inhibitors, where most of the colleagues are from the different departments of the university, and we also would like to see that AI/ML is utilized in this particular process. And I think the experts from some of the University of Hyderabad and many of these who have this experience could definitely help us in this. That's what I would expect. So maybe this will have to go further by a lot of brainstorming, a lot of things have to happen. So I think I have to finish now. I'm just seeing the time. With this, I would like to acknowledge all these with CSR, ICT, because National Cancer Institute, we have been extremely delighted, because I would say hundreds and hundreds of molecules were evaluated through them, including the semi-in vivo aspects on the hollow fiber aspects were done by them. Apart from the ACTREC, which is the Tata Cancer Institute's research hub, they have been wonderful collaborators for us in doing a lot of animal studies, the in vivo studies, because they are very essential before we go for some, part of preclinical aspects and so on and so forth. And many projects had come, no doubt, in-house and from various sources, including the Department of Biotechnology. So with this, I would like to thank you all. I think, this is a group photo I would like to show, because large number of them were involved in such programs, and I'm very happy that I had wonderful research students who contributed extensively, and that is how we are here today in this whole journey. So thank you very much, and my best wishes to all of you for this 2-day journey on this workshop. Thank you. And if you have any specific things, I'm ready to give you some clarifications or whatever you want. Maybe students should think of something.
So good morning, sir.
Good morning. You can introduce yourself and.
Sir, myself, Chandra Prakash.
Yeah.
I'm joined as a project associate.
Okay.
In this one industry. Sir, my question is that you have clearly explained PVDs, the pyridobenzodiazepines.
Yeah.
So actually, when we introduce that drugs directly, they have a lot of cytotoxic effects.
Yes.
When it is bind with the dimers, the cytotoxic will lessens. So my question is that recently Russian government has introduced a vaccine for cancer also, that is called as enteromics. So even though the vaccination is the best effective way to treat tumors, so why we are basically emphasizing on developing these kind of drugs apart from the vaccines?
See, vaccines is a different aspect. I will tell you, these drugs, today, one of the compound, what I showed you, the dimer, that particular compound is now, along with the ADC, FDA has given permission to go into the market, I think 3 years back. So they are already in the market, and they are for some specific cancers. See, you have to understand one thing. Cancer is not a single disease. There are hundreds and hundreds of these things in that, and each drug may not work for the other one. So you need drugs from all types of aspects. So this is one of the aspects, pyrrolobenzodiazepines, but there are a lot of other drugs which go through various other targets. It could be a pathway which is called as topoisomerase 1, topoisomerase 2. It could be tubulin polymerization inhibition. So PKI3, so many things are there. So here we have talk of one class, but people are working on almost all class of molecules. And here the success, as I said, has gone up to a drug which is in the market, which is being used in for clinical purposes. So I understand the general thing what you have to understand is we need drugs which can work on various targets, not just one. Just particular that, as you're giving an example of it will not function for every cancer. Okay, so because many cancers have different pathways, and for which suits, one has to go through an entire process, and there is a big, I would say it's a very big thing to do all at a time. You'll have to select one specific. Now people, for instance, work only on topoisomerase 1 or 2. Some people work as a target. So you took. Now people are looking for having molecules for 2 types of targets, so that whether the same molecule could work for 2 targets, that becomes beneficial sometimes. So these are the things. Okay, I think you understood what I'm trying to say. Okay. Yeah, please.
Sir, as you mentioned that dual targets, coming to dual targets in multi-targets, what are the aspects we have to keep in mind in the computational level?
Computational level, I'm not very 100% sure how to go about, because I'm not from that expertise. We'll be utilizing certain computational chemists on that. But the only thing is I can say is search for this can happen. Apart from the computational aspects, you can see the products, the compounds which are working on a specific target, and also compounds working on specific target. Then you know which is the active pharmacophore part in that molecule. Okay? And same thing in the other case. Then you try to bring these 2 pharmacophores together, bringing through certain type of a conjugation. How far and all that, there is a lot of hit and trial you'll have to see. Maybe in the computational aspects it may happen in some manner, but it is possible. It is possible through it will further give you an idea in the computational aspects, which may or may not work also. See, many computational predictions we have seen may not have worked. We have seen that also. Although, because again the computational aspects are based on several parameters. So even slightest difference in one of the parameters may not give you the right results. Because I'll tell you one example. See, when we were young, when the molecular modeling started, people thought that you can just make a drug immediately on the screen of the computer. But it's not the case. It cannot happen like that. Because you don't have that type of parameters to do in such a complex manner to see that. So slowly then people realize, no. Then what happened is the known drugs itself, you try to bring down the toxicity, you try to bring some better, improve the bioavailability or improve its effectiveness. Those things, computer modeling has definitely helped for the molecule. So to say that any 100% molecule has come through just molecular modeling, it's not very common. It's a very rare thing in 1 or 2 cases. That's because we also did libraries of something like hundreds and thousands of molecules from where we have shortlisted them, picked them, and then taken them further for actual this thing. So in some cases, there is some success rate, but it may not succeed everywhere, this aspect. Okay? Welcome.
Any other question?
This side, nobody. Girls. Okay, I think it's somebody, yeah.
Sir, you have shown that the pharmaceutical on the base molecule is 5 to benzodiazepines. So, sir, 2 units are required or only a single unit is required?
No. Maybe you have not understood it properly. In the pyrrolobenzodiazepine, you have certain aspects involved in that. One is the mean functionality, as I said, and there are some other aspects. So we have utilized to make them dimers to place where it can accommodate bulky things. Still, so that allows it to go inside the minor groove without getting obstructions. So why 2 were made, dimers and all this were tried were, because that is the one which has come today in the market, is to improve the base pair sequence selectivity. Okay? Because we were looking for sequence selectivity also in this while making such a thing. So that it has some selectivity with respect to normal and cancer cells. That is the main purpose. Okay? Or any other thing you have in mind?
Make a single drug, how many years will it take?
Yeah, as I mentioned in the beginning, it could take, in case of cancer, you have a little, you can do certain, I would say, fast track things. Still, unlike the many other normal diseases, it could take about 12 to 15 years minimum and could get extended also. So, because it's a very rigorous process, as you know. I don't know how much you have studied about the clinical, the drug discovery to development aspect, because it has, after the discovery can be about 3 years if you are doing properly. And then from there to preclinical. So if it succeeds in preclinical, you are okay, going for phase 1, 2, 3, 4. But if you don't succeed, you again go back to the discovery part and then try to bring certain changes in that molecule. So that is why you keep losing time. And then the best molecule, what you are thinking from all the parameters, the aspects, you take that forward. That is why many pharma companies, what they do is, particularly most multinationals, they will have at least 3 to 4 as backup. Because the one which fails, because many have failed at phase 1, phase 2, anywhere. So then they try to bring the other one or think of doing a change in the same one. So that is how it is done. Okay?
Any other question?
Nothing else. Please. Pardon?
Arvindo Pharma Limited.
What's your name?
So,
Lakshman. Okay, Lakshman. Tell me.
So, most of the molecules in piperazine are controlled. So, they may further degrade into formalin and impurities and may go reactive DNA. They are again the impurity converted into an carcinogen.
Pardon? No. It's not like that. See, the metabolism part, if you see, for cancer drugs, you need bioavailability if it is not getting through. Okay? Because many molecules, why they have failed is,
molecule state it is stable.
yeah.
When you are manufacturing
No, molecule also will not be stable, because once it gets into the biological system, a lot of metabolites are formed. So if the metabolite formation is such that your pharmacophore is getting totally disrupted, then it becomes useless. So till your pharmacophore is stable enough in the biological system to a certain time, then you get the effect. Okay? So as you must have seen now today, a large number of compounds have this piperazine moiety in many drugs to improve that. I mean, probably when we did, we hardly saw this. It was very rare. But today, a lot of molecules has come through this, where the improvement of biological activity has happened, having this piperazine moiety. So along with that, how the whole metabolism happens, we don't know exactly unless you study that. Okay? You cannot generalize just taking out piperazine separately and showing that it may not. It may have again with other functionalities, other groups. See, the main molecule thing may get into different pieces, which are, see, basically in metabolism, it's nothing but your body, I mean, the animal body or human bodies, enzymes, they function there, okay, to get the metabolites. Just like you think of, there was a big problem at one time, mostly a simple molecule like even aspirin or your paracetamol. See, aspirin has such a labile thing, you immediately get salicylic acid, even on storage for long. So that was a big problem. So that is why the formulation had to be such. If you get salicylic acid, it's no doubt it is an active molecule, but to the body it doesn't do well immediately. So that is why these things have to be studied. You're very difficult to just say like that, such a group is like this. No, you can't say, because how the whole thing happens, along with the whole structure of that you have to see. What you say may happen in some cases. I'm not denying that. Okay?
Any more?
Okay.
Very much, sir, for this overall view of medicinal chemistry and how we can think of creating a drug molecule. Just look at the molecule. I understood like this. Just look at the molecule carefully. What are its components and how can we think of its preparation or synthesis, and then what groups will affect the toxicity of the molecule, how we have to avoid it, and how to connect it, how to bring that molecule into DNA and then make it interact with and give a better life to the human. So thank you very much, sir, for giving us a picture, and then thank you, sir.